Okay, I think we can get started. Welcome to all who have joined us. I'm Michael Bruno, Provost of the University of Hawaii at Mānoa, and I am honored to host this important and timely webinar, 12 months to 12 weeks, how a UH Mānoa lab is fighting a global pandemic in Hawaii. This webinar is part of a larger series that we've titled Community Conversations. The series will feature informative conversations with University of Hawaii at Manoa subject matter experts who will share relevant and valuable knowledge with our community. The series also aims to inform and inspire students, alumni, donors, and members of the broader UH community. I am proud to work alongside some of the world's leading scholars in a multitude of domains, and I have never been prouder to be a part of the UH Mānoa Ohana than over the last 6 months, as I've watched our faculty and staff actively engage across Hawaii in so many critical areas related to the health and well-being, economy, and social justice and equity of our home. These faculty and staff are also working tirelessly to ensure that our campus can safely reopen, albeit in a reduced density fashion, for the fall semester. I want to thank each and every one of them. Mahalo. Joining me today are two of those faculty experts who have poured themselves into assisting the broader Hawaii community as well as our campus, Ohana. Professor Vivek R. Narukar joined the University of Hawaii at Manoa in 1994 to develop the Research Centers in Minority Institutions program, a National Center for Research Resources funded retrovirology activity. This followed a 5-year appointment as a visiting fellow and visiting associate at the National Institutes for Health. His major area of research is in infectious diseases, specifically the study of pathogenesis of orphan diseases and orphan microbial agents. Over the past 2 decades, he has conducted research in the diverse but related areas of virology, specifically neurovirology. Professor Rosie Allegado was born and raised on Oahu. She is an associate professor of oceanography and Sea Grant, where she is the director for the Center of Excellence in Integrated Knowledge Systems and a member of the Center for Microbial Oceanography Research and Education. Rosie completed her postdoctoral work in evolutionary biology at UC Berkeley and holds a PhD in microbiology and immunology from Stanford and a BS in biology with a minor in environmental health and toxicology from MIT. Her work focuses on investigating how microbes shape the adaptive potential of their ecosystem across a broad range of biological and temporal scales. She is deeply committed to increasing the participation of underrepresented minorities in STEM, and she is the director of SOAS's Mile Mentoring Bridge Program. In 2018, Rosie was confirmed to the City and County of Honolulu Climate Change Committee. Without further ado, Rosie and Vivek, the floor is yours.
Aloha. Thank you, Michael, for the opportunity to share our work. We're really excited to share what we've been up to and to answer community questions.
Thank you, Michael. This is great. We will be able to share with the general community what we have been doing here at the Tropical Medicine Clinical Lab at the John Avon School of Medicine here. We are very excited, and the title of the presentation is very interesting. And people have asked me the question, what does it mean? And we are going to try to define that and let you know.
So right now I'm going to share my screen so we can talk about a little bit of what we've been up to. I'm going to work on playing my presentation. Can you all see it? Looks good? Yes, okay. So how we've been fighting the pandemic at the University of Hawaii Manoa is through the establishment of a brand new clinical lab, which we call the Tropical Medicine Clinical Lab at the John A. Burns School of Medicine. So I think it's really important to first talk about what is a clinical lab. And we're going to define some terms because at UH we have lots of different kinds of labs, and across Hawaii we have lots of different kinds of labs for different purposes. So a clinical lab is a healthcare facility that provides many different lab procedures that aid physicians in carrying out diagnosis, treatment, and management of patients. I think all of us have gone to the doctor and had our blood drawn or provided some kind of fluid sample and, you know, gotten the test results back, right? So we're talking things like everything from diabetes to blood sugar levels to pregnancy tests. That's the kind of thing that a clinical lab does. There's a difference though between a clinical lab and a public health lab. Both laboratories perform many of the same testing procedures, but their patients and who they serve are very different. So clinical labs are assisting clinicians or physicians with individual patients, a single patient. So an example of this would be diagnostic laboratory services or clinical lab services of Hawaii. And there are more. This is just a couple of examples that you might be familiar with. A public health laboratory like the Hawaii State Department of Health that we've been hearing from when we hear those news broadcasts, those laboratories are supporting the health officer whose patient is the whole community. So in the case of the COVID crisis, our public health officer has been Dr. Sarah Park, and it's been supported by the state diagnostic labs. And Vivek is going to talk about the difference between a clinical lab, which is what we have been establishing, and a research lab, which many of you might think of and associate with the University of Hawaii.
Right, and that brings us to the point of research labs. You know, we have research labs all over UH and here at the medical school. The research lab usually functions in creating new knowledge and new research activities. So as Rosie mentioned, clinical labs scientists diagnose, monitor, and treat diseases. But when the testing is done, all of this testing is done in a research lab originally. And that original research lab testing protocols are developed and then passed out to the clinical labs. So here in the research lab, the scientists investigate the study, the health and the illness of people and answer questions about medicine and behavior. We test different protocols, and these protocols are then later fine-tuned and put into a clinical lab to do testing, such as the COVID-19 testing.
Right. And Vivek, correct me if I'm wrong, but the Jabson Tropical Medicine Clinical Lab is the first clinical lab to be established at UH Manoa. Is that right?
You're right. So this is the first ever lab to be established in here, and we'll be talking more about this lab and what is the function of this lab and how will this lab serve the community.
Right. So you might ask yourselves, why does Hawaii need another clinical lab? Because as I stated, we have some really amazing commercial labs and private labs available. The first is because we're in a global pandemic and we need to have all hands on deck. And we need to build the capacity in case we see an increase in infections like we are now. So we feel that UH bringing on another clinical lab will support the state of Hawaii's Department of Health, hospitals, and other existing clinical labs so that we can help identify positive disease cases. The second reason has to do with the fact that we just talked about now is the connection between research at the University of Hawaii and clinical labs. It's the research partnerships, and Vivek's going to talk more about it.
Right, and again, for research partnership, we are a research university, R1 classified research university, and our main goal is to focus on research. But at the same time, for conducting research, you also need to do many times clinical work, and that clinical work requires obtaining sample specimens to understand what the patient is going through. So this particular lab can be an interface between our researchers here at UH and the laboratory, in which we can develop and validate new diagnostic technologies. We can identify recovered patients whose sera can help others. We can do some of these testings in here to understand are these patient serum samples good enough to be used in treatment of patients?
Yeah, I think that's a really, really critical aspect that makes the JABSOM Tropical Medicine Clinical Lab, which we call the TMC, it makes the TMC very uniquely positioned because we're right at that interface between what's going on in the clinics and what's the research that's going on in the labs. And because we're at the UH and we're made up of faculty researchers, it's very easy for us to talk to each other and break down any kind of any barriers to communication. So what does it take to start a lab? And don't be scared by this crazy diagram. So let's start in the middle. The first thing you have to know is that if you want to start a COVID-19 CLIA lab, you have to do a lot of work. The first thing you have to know is that the lab has to be what's called CLIA or CLIA compliant. And CLIA compliant, I'm going to read just a brief so that you guys know, I think it's important, stands for Clinical Laboratory Improvement Amendments. And Congress basically has established quality standards for lab testing for any specimens from humans, like fluid and waste, and different kinds of tissue and things like that for the diagnostic prevention of disease. So the first thing is any clinical lab has to be able to meet these requirements, and there's many. The next is that the COVID-19 test, when you get something stuck up your nose, it's quite a high complexity test. It's more complicated than many of the blood tests that you might have taken in the past. So that falls under the category of high complexity testing, and that has additional requirements that we would have to fulfill. So Vivek, do you want to take us through some of the specific requirements on the right-hand side of the screen?
Right. So I think as you see that these high complexity CLIA labs, they are not easy to develop and establish. When we started doing this work in the month of March, we had to identify a laboratory director, whom we can see on the top in there. And the laboratory director's function is to approve each and every test result from the lab. It's very important that this tests are being done efficiently as well as correctly as per the CLIA compliance. And the laboratory director will then communicate those tests with the attending physicians. There has to be a technical consultant, he again, an MD level technical consultant who can provide consultation for each specialty test in the lab. We have clinical consultants who can address questions, concerns from physicians and patients about test results. It's important that these consultants are in continuous discussions with the lab director as well as the technical consultants. And then most importantly, we need to have, if you want to do some kind of a testing, we need to have a laboratory space, which is the biosafety level 2 plus laboratory place. Especially for COVID-19 testing, we need to have a high containment kind of a lab where we can do this test routinely. And all of these tests which we do, they must be FDA authorized tests. So what does that mean? That is the Food and Drug Administration along with the CDC, they have a set of tests which they have on the market. And those tests needs to be done, only those tests can be done in a CLIA certified lab. Currently, we'll be doing a nucleic acid-based testing for the virus, as well as the antibody testing of the blood. And then the testing personnel, who are the heart of this whole thing, these are really competent technical staff who will be doing routinely all of these tests. It's important that our technical personnel staff are very well trained too.
Not only that, each of these personnel, the lab director, the technical consultant, the clinical consultant, and the testing personnel have to be licensed by the state of Hawaii, and they have to be current. So it's not only expertise, it's licensure. Now, that sounds almost impossible to pick up some random people off the street and do that, but because we're at the University of Hawaii at Mānoa, and I think that's what's so amazing, is we not only have the resources needed, we already had the people in place. And that is what made this opportunity, this need that we met with this opportunity to form this lab so unique, is that when I have to admit, I was very naive and I was like, I saw this title and I said, lab director, okay, one of us can be that. No, I had to learn a lot. And I learned that these positions, it requires a lot. And I was so impressed that at University of Hawaii at Manoa, we actually are able to fulfill all of these needs, and we have these resources to do this. So that really gave us confidence that we could be able to meet the needs of the people of Hawaii. So how did we do it? This is a timeline that I'm showing you here. And so at the very bottom, you can see that we are trying to do this in phases. Phase 1 is we just have to establish a physical kind of brick and mortar clinical lab. And then we plan to accept samples in partnership with community healthcare centers. And there are 7 community healthcare centers on the island of Oahu. If you're wondering like what is a community health center, that would be like Kokua Kalihi Valley, Kalihi Palama, Waikiki, all the way out to the Waianae Comp, and also Waimanalo, and anywhere in between. So there's 7 across the island of Oahu. So we're going to do phase 2 is going to be the nucleic acid RT-PCR testing, and then we're going to phase in for phase 3. Now, when I say phase 2, that doesn't mean we're going to stop. It just means that's the phase that we're going to begin to work on those. So way back in March, actually, I remember the date. It was Friday the 13th, March 13th. Vivek and I had been emailing each other. And of course, I was just super naive and very enthusiastic. And I said, we can do this. And Vivek said, there are a lot of things we have to get together. And the very first was to identify the SL2 lab space. Vivek, do you want to talk about the team that you had been able to put together?
Right. So, having a CLIA lab was a dream. And when I saw this opportunity that we can do it in here, and with help from Rosie and others too, we put a team together. And probably we're going to, the next slide, we're going to show our team. Having the members of this team in and identify them that they are the right people who can do the job. And I was very impressed to see that all of those particular positions, the people whom we need, they were here at UH Manoa and or at JAPSOM in here. And when I approached them one by one, every single person told, yes, we are on board. Just they tell us what to do. And that kind of gave us more confidence in saying that, okay, we can do it, because after all, workforce is more important than anything else. And then, of course, there was the need of the space where to do it. So I had to approach the dean of our school, Dr. Jerris Hedges, and he was very kind enough to say, yes, we can. If we can do it, we will find some space for that too. So we put together a CLIA application really fast, almost within a matter of a week or so. And once that application was done, Rosie physically took that application down to Kapolei and submitted the application. Maybe Rosie can talk more about that.
Well, yes, because we were unclear at that point. If you remember, if we go back to March 26th, that was the week that everything started shutting down in Hawaii. It was unclear whether or not we were going to be able to get a courier to come and pick up our application and take it to the state DOH office that we needed to take it to. And so after much going back and forth, I just said, I am going to come and pick up the application and drive it myself. And yeah, that was like a very big adventure to go. I'll tell you what was really crazy was I picked up the application at 2 p.m. By the time I got it to Kapolei at 3 p.m., they had started making the announcements that they were shutting down the state of Hawaii. So we just felt like we really, really we felt so motivated to do this for our local communities. Do you want to go on?
And then, yeah, and then within again, we were surprised by April 1st, we already got our CLIA licensure. And that gave us the more confidence that yes, we can do it. And that just put in the whole ball rolling about asking for funding. And believe me, we went to many organizations, including the UH as well as state and the city, asking for funds to do this kind of a project. Because now we had a team in place. The question was, who is going to fund this project? And maybe Rosie can talk more about the funding coming from the city and county.
Yeah, so the funding that comes from the city and county of Honolulu is part of the National CARES package. So it's part of the money that the federal government has designated to help us fight this pandemic. And we through our connections with the city and county, we basically put together a proposal. And this was a huge learning process because Vivek and I are scientists, but we're not like business people, but we were learning on the fly. And so you'll see in a couple of slides where we thank numerous on campus and off-campus entities who just really help teach us how to put together these kinds of proposals so that we can, short and sweet. Vivek and I think are used to putting together 25-page research proposals, and we learned to put together 2-page research proposals, and they weren't proposals, they were prospectuses. So we were able to do that. And I think one thing that was really amazing is that Mayor Caldwell and his team were extremely amazing with the foresight to see that we would need in the fall, or maybe even in summer where we are now, the ability to increase capacity, and specifically to provide services for the people of Oahu who might not necessarily have access to this kind of testing. So this is a really wonderful partnership between the community healthcare centers who are charged with helping our people across different socioeconomic capacities and who have different abilities. Maybe they don't have health insurance or for various different reasons. We're going to be able to specifically help to serve these underserved. So it's a very nice complementing, because you have the private labs and the clinical labs who are serving those who have insurance, and this will help because the city is going to be paying for those tests. Vivek, do you want to go on and finish?
And then we had to go through some bumps up and down, and finally the UH Board of Regents approved our whole proposal and protocol, and finally we just got the funds released from the city and county. I want to take a pause in here and thank the administration of Mayor Caldwell as well as Mayor himself for giving us this funding.
Yeah.
So the next question always comes in that, what kind of a test we are going to be doing, and we will be talking to you about that more in the next slides.
Yeah, and so just to say that where we're at now is we're moving at light speed. We're moving so fast. We have identified and qualified testing personnel. We've ordered the machines, and so we're speeding as far ahead as possible. Okay, so what kind of tests will be performed? What's going to happen is that somebody will go to a community healthcare center, and that medical provider at the community healthcare center will collect a sample or specimen and send it to our lab. And so we will be going around and collecting samples on a set schedule from each of the community healthcare centers, like every day, pretty much. Those samples will be prepared. What we're going to do is we're going to bust open the virus and we're going to isolate its genetic material. And we use these machines that help us to do many samples at once. It's called automation. And what we do is we're going to extract the genetic material, and the genetic material of COVID-19 is RNA. And then what we do is we quantify it. We count how much was in that sample. So Vivek, do you want to talk a little bit more about the type of tests we're going to be using?
So we'll be using a polymerase chain reaction technology, or PCR-based technology. This is the unique technology. There are different forms of this technology. We'll be using the technology which has been developed by Thermo Fisher using the Applied Biosystems machines. Now, I must say in here that you must be hearing on the news very recently, rather as of last 16 hours, that there is a shortage of these reagents and reagents for doing this test. It's important to know that right in the beginning, in month of March itself, we were looking around to see who has what kind of technologies here in Hawaii. And when we saw that we need to use a technology which is not currently available on the island in here. Why? Because if there is a problem with the supply chain, we all don't get stuck with it. So our technology, which we are going to be using, is not a Roche technology. It's a Thermo Fisher technology. So, and currently the reagents are available for these kinds of technology.
Yeah, so really important to remember is that the Thermo Fisher test platform is completely FDA approved. It has FDA authorization. All the tests have FDA authorization. In fact, one of the main reasons why is, of course, because we wouldn't have to have any concern about any of the federal approvals surrounding these tests. And as Vivek said, it's really important to have a diversified kind of testing landscape so that, Hawaii is at the end of a long supply chain. So it's good for us to have options so that if one lab can't, get the supplies, another lab can come and help. And that's what surge capacity and being complementary is all about. So once the test is performed, a report is put out, and as you now know, the lab director is going to certify that. We're going to be sending those results to the community healthcare provider and the Hawaii Department of Health. And we're working with the UH Health Partners of Hawaii, University Health Partners of Hawaii, to develop a HIPAA secure, so that the patient information is secure way to transmit this information. Oops. And then of course, the DOH will notify the public and begin contact tracing if appropriate. So it's very, very similar to everything that's currently going on now. I would say one of the big differences is that we have chosen the Thermo Fisher platform. Do you want to add anything else, Vivek?
No, I think you got everything. Good.
Okay, so we want to really emphasize that even though Vivek and I have been getting a lot of attention, here's Vivek in the lab, and here's me hiking with my kids, that this was a team, it was a team effort. In Hawaii, we say it's a kako thing. We could not have done this without the efforts of all of the people at UH, our faculty, staff, and students who really committed the time and effort to making this possible. So we have Mike Lieberman, who is our lab director, David Horio, who is our clinical consultant, Vivek's actually our technical consultant. My job is actually to interface with the community centers. We have Lauren Ching, who's a graduate student, who has been absolutely essential in helping us to understand procurement and supply chains. Katie Hinson, who works with me at Sea Grant, has been absolutely essential in helping with our communication with our community partners. Sandra Chang has been our logistical foundation, helping us to make sure that our website is up and running, that all of our certifications and personnel hiring are going smoothly. And we have Eileen Nakano. I actually have to apologize. That's not Eileen's picture. I accidentally put the wrong picture in. But you will see, we actually have a video at the end of Eileen, at the end of this presentation that you'll be able to take a look at. But Eileen has been completely essential at being our lab supervisor. Vivek, do you want to say anything else?
I think without this team and many more, we are just having these few people in here who have been the backbone of putting this lab together. But there are many people behind the scene who are with the JAPSOM administration, UH administration, Office of General Counsel, Office of Research Services, ORC, Office of Research Compliance. They have been in the loop 24/7, and they have provided us so much support. Without their support, we would not have been here where we are today.
Yeah, we definitely want to recognize our campus and community partners. I mean, UHC Grant was very, very supportive, number one, in allowing me to do something that was outside of my wheelhouse and my job duties, and enabling me to really seeing the passion and actually how this would benefit our coastal communities to work on this. University Health Partners, who, again, as I said, are providing our HIPAA secure reporting and personnel. We cannot thank all of the staff in the Office of Research Services. This is the kind of thing we never get the chance to do. And we were really emphatic that we needed to acknowledge everybody who really helped to push this. And, the reason why we could do this in 12 weeks is because we had these unsung people who believed in this and really helped to carry this forward. We couldn't have done this without the Office of General Counsel. I really want to particularly point out Presley Pang for making sure that all of our T's are crossed and our I's are dotted. Of course, UH Foundation for helping us to find support. JABSOM, as Vivek brought up, the Office of Sustainability, the individuals in that office really helped us to really hone our proposal message. Of course, we're looking forward and continuing to work with the Department of Health. Vivek and I send emails to the Department of Health maybe like 3 or 4 times a day, making sure that our progress is moving apace. The Hawaii Primary Care Association is the medical provider for the community healthcare centers. And we really have to shout out the people and organizations who are supporting and paying for this effort. The Rockefeller Foundation contributed the funds so that we could buy the machines that we are going to be using. And a huge bulk of the support came from the city and county of Honolulu. And I really can't emphasize enough that the people of the city and county are really caring and looking out for the people of Oahu. And we look forward to continuing to work with them. Vivek, do you have anything else to say?
No, I think we have covered almost all of our partners. I do want to say thank you to the city and county, as well as the Rockefeller Foundation. Rockefeller Foundation, all the way from New York, we had a good discussions with them, and they are funding a lot of different agencies, and we are proud to say that we will be funded by Rockefeller Foundation.
Yeah. So that ends our slideshow. I'm just going to exit out, and I'm going to stop sharing my screen. Okay.
I do have to unmute. You'd figure that I'd know how to do that by now. Well, guys, that was fantastic. And as always, the two of you are very generous in thanking your colleagues and the supportive organizations. So much appreciated as well. But I will take the opportunity to thank the two of you for being unyielding and tenacious. And you saw right from the beginning, this had to be done, and you did it. So maybe I'm going to ask the first question, and maybe that's something that our well over 100 viewers are wondering. The title, 12 months to 12 weeks. There's obviously deep meaning in that title. So maybe just a few words about that.
Maybe I can just take a few things, and then probably Rosie can follow me on that. So basically, any of these kinds of efforts takes over 12 months to actually establish a lab and do a lab. I say up to 18 months, not only 12 months. It's a long process. And as I told you earlier, that people who are involved in this, right from not only the team which we have built, but also the people behind the scene, because of them, we were able to put this together in 12 weeks. And that's where the 12 weeks to 12 months comes into the picture. So this particular process, and many of the who are in the field will tell, this is amazing, because it just does not happen. But remember, this is a marathon. This is a marathon for everybody. When I remember when I talked to Presley Pang at OGC, he said, this is not only a marathon, this is a marathon every single day for me. So everybody was working very hard. So that's where the 12 weeks to 12 months, 12 months to 12 weeks comes into picture. Rosie?
Yeah, part of it is that we did not work regular business hours. We worked nonstop, day, night, weekends. I mean, one group that we need to actually really recognize is our families. Our families bore a huge sacrifice so that we could continue to work on this. I was teaching 2 classes. Vivek was also teaching classes in the spring semester. We both have an active research lab with our graduate students. So we need to thank our graduate students also for being so independent and strong. And of course, our families for being able to manage without us. But yeah, I think, when we saw what was happening across the world, and we know that Hawaii is very isolated, we knew that we had to do something. We couldn't wait for someone else to give us the solutions. And what's really remarkable is that the reason why we could do this is because UH has such a deep bench. We have an incredibly deep bench of talent. And that's something that I learned. And the talent isn't just in the academic prowess of our researchers. It's in the hard work of the everyday rank and file staff at campus. So I just am so thankful of all the work that everybody put in.
I just want to add one thing is that, so our graduate students and our staff, I remember calling them over the weekends and the long weekend holidays, and they were sitting with me on a Zoom. I don't know whether Zoom is good or bad, but at that time, Zoom was available and everybody was on the Zoom. Myself, Rosie, and our graduate students, and talking about how we are going to go further, developing the budgets. But this is all happening over the long weekends.
Yeah.
I think the only day we took off was Mother's Day. That is literally the only day we took off in the past 3 months. Yeah.
Well, thank you again. So some of the questions that we've received from the audience. One, I think you've already touched on, will the lab be doing COVID testing for the community? You've answered that, but then a second part to that question, will it be made available for private and or business use, which is an interesting thought.
Yeah, Rosie?
Yeah, so currently through our partnership with the community healthcare centers, we are going through that process. However, with our partnership with UHP, we can set up a way to get billing. And so we are totally open to having partnerships with the private sector. Yeah, that's totally possible. And that information is on our website. So if you just Google JABSOM Tropical Medicine Clinical Lab, you can see, I think we have a section called services, and we do say that we are open to partnerships with the private sector.
Great, thank you. So here's a question I think that is on the minds of many people. We read a lot about the different testing that is out there, two mainly, right? The genetic-based PCR testing and the antibody tests. Can you give some comments, some insights to the benefits versus costs of each type of test, maybe a bit about their accuracy?
Sure, I'll get started. And then Vivek, is that okay? Okay.
Yep.
So I think the most important thing to think about when you think about cost versus benefits is why do I need to get this test, right? So if your question is, do I have the virus? Am I infected? Then that's an individual test, and you want to get the nucleic acid RT-PCR test. That's the one that's like up your nose, because that will tell you if you have an active infection. And of course, its accuracy depends on whether or not in your body, you have the amount of virus that will be detectable. It's a very sensitive test, but if you're at the very early beginning stages of the infection, your body may not your body may be effectively fighting the virus, and it might not be at levels which this test can provide. Now, a second reason why a different question might be, how much exposure is there in my community? So for that question, you might use a combination of the RT-PCR test as well as an antibody test, because the antibody test measures how many people in the community have been exposed to the extent where their body mounted a good enough immune response. So again, the accuracy of this has to do with, can we measure or detect these antibodies in their blood? And so for that question, that's an important question, because that can tell us the prevalence in a community. That might help us make some policy decisions as far as costs and benefits. And then a third potential reason for costs and benefits of this would be, am I somebody who works in a high risk, high exposure position? Am I working at the hospital, or am I working in a nursing home, perhaps the prisons, or even perhaps the airport in the case of Hawaii and tourism? So in these high exposure things, somebody might want to be tested frequently in order to assure that they don't bring it home to their families. And so for that, you might be wanting to use a rapid test. And so Vivek can speak more to the exact numbers about that. But it's really important to establish why you need the test.
Right. I think once you establish why you need the test, the question is that not all tests are same. Okay, there are various different kinds of tests. We have to look at the specificity and sensitivity of these tests too. Now, Hawaii did not have that many cases, so it's more important that we need to have a very highly specific and highly sensitive test, which can really tell that the person is positive or negative. If you go to New York City, a couple of weeks ago, the infection rate was really skyrocketing. At that time, you may not need that kind of a highly specific and highly sensitive. We need to see whether who is positive in there too. But the main thing which I want to draw back is people want to see what kind of antibody test. Everybody's talking about immunity passports. The immunity passport thing is getting a lot of press. You need to understand that not all serological tests are made same either. So we really need to define what kind of a serological test we are going to be having, which can detect the antibodies. But just because you have an antibody in your body, in your system, that does not mean that you are protected. This is a false kind of a security that you have been protected, because SARS coronavirus 2 antibody levels in the human being, they wane off. Okay, and again the question becomes is that how much antibodies you can have. And that's something which we can do, and what we call that as a neutralizing antibody. So the virus can be killed by this neutralizing antibody. Just you having an antibody that is IgG positive is not good enough. You need to have neutralizing antibodies. And those kinds of neutralizing antibody tests, something which we can do here at JABSOM, because we have a biocontainment facility. And in that facility, we can actually do the titers of this neutralizing antibody. And that is something which we'll be doing in our phase 3 of this, to make sure that we can tell the physicians that this particular sample which you have collected from a patient who has recovered from COVID-19 is a very good sample for using that for another patients. So that's something, a capacity which we have here as a research capacity, as well as we can use it as a CLIA compliant assay.
Well, thank you both. That was very enlightening. So, kind of building on that, so obviously, to the value of having antibodies, of course, all eyes are on the development of a vaccine, or perhaps a set of vaccines. So another question that came in that kind of starts broad and then really drills down, interestingly. What is the status of vaccine development for COVID-19? Second part, are there any vaccine testing trials here in Hawaii? And the last part, do you need volunteers?
Very good questions. So vaccines, yes, people have been waiting for this vaccine, and to hoping that there'll be some kind of a vaccine where we all can take one and go back to work and start the economy, basically. And the vaccines are not easy to do. Previously, used to take up to 15 years to get any kind of a therapeutic drug or a vaccine off from the FDA and into the market. With very newer technologies, we have seen during Ebola outbreaks and Zika virus outbreak that we were able to cut it down to 18 months or so. And that's what right now the government as well as the FDA is saying, that we can have a vaccine probably in next 12 to 18 months. If you would have heard some news yesterday, there have been centers opened up now where they are accepting volunteers for future COVID-19 vaccine test. Now, for human trials, we need vaccine designs that could be rapidly produced in a very large amount, that is millions or billions of doses. And we all recognize that an effective vaccine is the best solution to the COVID-19 pandemic. But safety and efficacy are two important properties of a vaccine, or used by billions of people globally. These first generation vaccines include one basis on mRNA and non-replicating adenovirus vectors. DNA vaccines are also being developed, as they too can be designed and produced rapidly. The SARS-CoV-2 immunogen expressed by these vaccines is a spike protein, or S protein, the sole viral target for neutralizing antibodies. As I mentioned to you earlier, neutralizing antibodies are very important, but the vaccine can produce neutralizing antibodies against the S protein, and we can use specific adjuvants along with that too. So the goal is to induce a neutralizing antibody titer after 1 or 2 doses of the vaccines that can either protect against infection, that means you can get a sterilizing immunity, what we call, or reduce the severity of the disease in any recipient who still becomes infected, or they can also prevent transmission of the virus. And the next question was asked about, do we have anything going on at UH? Definitely. At UH, we are developing SARS coronavirus 2 spike protein-based subunit vaccine using novel adjuvants. We have exciting preliminary data using small animal models. We have applied for BARDA and NIH funding. We are collaborating with biotech companies in Hawaii, such as the Hawaii Biotech, and nationally with companies like Sologenics. And we have a history of collaboration on various vaccines for dengue and Ebola with this company. Basically, we are in the preclinical trial phase. Hopefully, within the next few weeks to months, you'll be able to hear what we have been doing. Our researchers who have been tirelessly working on vaccines will be giving some information out to the community over the next couple of weeks. Rosie?
Yeah, I mean, no, I think Vivek is the expert. He summed it up really nicely. I mean, I think the main thing to take away from this is, yes, University of Hawaii is working with our small but very robust biotech community here in Hawaii to develop. And so we, again, the main thing you should get is that we aren't waiting for other solutions, because we know that our ideas, well, number one, it's very important to have a diversity of options for vaccines, and for actually any research. It's always important to have a diversity of people working on a really big problem. And so we're saying that we have something to contribute to this really big problem in terms of problem solving, and that's what we're doing. We're part of the solution.
So, and building right on that, Rosie, I think it's very clear to everyone watching how passionate the two of you are in serving the community and contributing to the health and welfare of that community, and maybe even exporting what we learn beyond Hawaii. You are researchers at heart. So does this new capacity, which is certainly something unique to the University of Hawaii at Mānoa, does it form the foundation for expanded capabilities? We know this is not the only pandemic that we're going to face, very likely. But then there are other diseases. Vivek mentioned the work in dengue, and we have other work going on in both in Hawaii and other parts of the world. Does this lab strengthen? Is it a new thing for us as a tool?
Right. Let me take that question, because we talked about the interface between clinical labs and the research space. I must emphasize that these two are really different things. And they should be different for management of patients. But there is an interface between that. We expect that COVID-19 to be a thing of the past over the next 2 years or so, based upon what we saw 100 years ago. With the Spanish flu, it lasted for almost 2 to 3 years, and then probably nobody talked about that anything anymore. And we are hoping that will happen over the next two years or so. So when that happens, the tropical medicine clinical lab will conduct tests on something called neglected tropical diseases that affect the people of Hawaii the most. And some of these diseases, really speaking, I need to be open and frank in here that NIH does not much support that, because they are not affecting thousands and millions of people in here. They are affecting few hundred people, or maybe few thousand people here in Hawaii. Some of them are dengue, Zika, rat lungworm disease, which we hear a lot from the Big Island, murine typhus, toxoplasmosis, and leptospirosis. These are 6 major diseases which are affected here in the islands, and our community is the focus of all of those diseases. And we are poised to assist the Department of Health in conducting tests for these diseases in a CLIA certified lab. For example, for dengue, it's sometimes necessary to conduct plaque reduction neutralization tests, or PRNT, at UH Manoa. At UH Manoa, we have been doing this thing for ages, almost 50 years now. So we can easily transfer this technology into a CLIA lab and have a print kind of a technology developed in here too. So yes, this laboratory will be used for other diseases which affect the people of Hawaii.
If I can build off of that, I, as a member of the Climate Change Commission and a microbiologist, something that we know is likely to happen in the future is that as the planet warms, as the Pacific warms, we're going to see new and emerging infectious diseases. And so first of all, the Tropical Medicine Clinical Lab is going to be perfectly positioned because of its strength in tropical medicine, together with its ability to kind of really, be on that cutting edge of monitoring and seeing what comes next for not just Hawaii, but for our other Pacific Island, nations and collaborators. But I think it's also important because this will provide an opportunity for training future leaders beyond just people who are going to get MDs. I think that there are many career paths that can include JABSM, but beyond, we're looking at the need for increasing medical technologists, nursing. I think it's so important that we continue to train our own public health staff and build that capacity, because Hawaii, we face unique diseases to the continent. Our unique ethnic backgrounds, we have unique issues and health disparities that we can build upon and work in partnership. The JABSM has this amazing Department of Native Hawaiian Health, and through that they do a lot of really amazing community-based participatory research. And so we can build those partnerships with those units within JABSM to begin to really drill in on some of those. There's biomedical engineering. I think one thing that we've seen is that we have been innovating at University of Hawaii, building our own masks, coming up with ways to make cloth masks, not just face shields. So there's many opportunities for advancing and creating, technology. And then as far as research, I think what we're seeing is that when we have cross-unit collaboration, we can really expand. So somebody from oceanography can work with somebody from JABSOM, and what unites us is microbes. So we have, just to give a short plug, we have an amazing cohort of emerging young and now established investigators in Simaiki, which I hope you'll invite those people to come and talk too. But oceans and human health is a huge emerging topic, and there's nobody better than the University of Hawaii Oceanography department to begin to ask those questions. And it's not just that, we have strength in many different areas, and I think we need to own that and see how we can inform what's going on elsewhere in the world. We have the solutions that other people can really benefit from.
So I'm going to jump on that thought. You just used the word inform. So we both work in areas related to climate change. Some of us have seen that science and that issue be turned into a political issue, very sadly, right? And so I recall one of the questions, and we had a conversation about it, was related to the level of trust in science in Hawaii. And I think back to the early days of climate change and often think about what did we do wrong as a science community in messaging the reality and the urgency. So likewise, we are looking at a ramp up towards vaccine development and distribution. There are already concerns, even protests about vaccination, efficacy, safety. Do you see this enterprise that the two of you and your teams have embarked on as also an opportunity for public education related to both mitigation, avoidance, and also vaccination?
Yeah, so I'll start and Vivek can finish. I think that it's incredibly important for us always to remember and acknowledge that we as clinicians, we as scientists are part of the community. We're serving the community, and it's incredibly important that we continue to communicate and engage and authentically engage and receive input and feedback. If the community is concerned about vaccines, that is telling us that we need to do a better job at communicating the importance of those vaccines. It's telling us that maybe the way that we have, made legislative policy or other kinds of policies raises important concerns that, sometimes I think a lot of times when we try and come up with a solution, the solution creates new problems, and we have to be really cognizant at listening to our local communities. So yeah, I think this is an opportunity to listen, because our communities want to be safe too. And so if we can find a way to partner with our communities and have our communities leading the way, I think that could be extremely powerful.
I just want to add quickly on that, because don't forget the JAPSOM Tropical Medicine Initiative. So we started an initiative, let me not use the word we here, our graduate students started an initiative 6 years ago called as Student Immunization Initiative, SII. It's fully run by the graduate students. We are there to help them out. And they are the ones who are promoting vaccination and vaccines specifically for HPV among young girls and boys. So our folks go to the local middle schools and high schools and talk to them about vaccination, hoping that the rates of HPV vaccination, human papillomavirus vaccination, will be going up. Dr. Sandra Chang, who is one of the members of our team, she does this all the time. She works with the Department of Health and goes to the high schools and trains these people, hoping that constant learning process will at least get that to the home, because after all, it's the parents who are going to say yes or no for the vaccination. So we have a student immunization initiative, and we work very, very closely with the local communities, specifically the high schools and the middle schools.
Oh, that's great. Thank you both. So speaking of thanks, in our last few minutes, it's my time to offer thanks. First of all, to the two of you for taking time out of your obviously busy schedules and be with us today, and for the amazing work that you're continuing to do. And to your team, I'm sure all of whom are watching, a big mahalo. I want to also acknowledge the donors to the COVID-19 and Infectious and Emerging Diseases Fund. This was a fund that was created some time ago and during the early days of the COVID pandemic, when we really did not understand what was emerging and the threat that it posed, and even how we were going to best utilize those funds at Mānoa. Still, even in those early days, the community came together. They were extremely generous and quick to donate to this important fund, which I hope our donors recognize has been put to extraordinarily good use as we at the university continue to develop local solutions to this ever-changing situation, and hopefully export those solutions around the nation and around the world. And that is our hope. And with that hope and that belief in you and your teams, we'll say a big mahalo and say to everybody watching, thank you for joining us, and stay safe, stay well, everybody. Mahalo.
Thank you.
Thank
Mahalo.
you. Aloha, everybody.